the first question was about individual-based models. It was actually about population level, which is not quite the topic we're doing here. We're doing within host, but I figured I'll pull one up anyway. And also because this specific one has a multi-scale model where they're running a within host model inside the agent-based model, and that came up already. So I figured that could be interesting to briefly look at. So this is the paper here. And I'll put those papers in that zip file with all the other papers, so you can download them at some point. So immunoepidemiological, that's what they're calling here. So sometimes they call it multi-scale. This term I haven't seen that often, but it's very descriptive. So they're having an epi population level model with some immunology on top of it. And of course, we can't work through the whole thing in a few minutes, but here you see, they use different letters, but it's kind of sort of similar to the models we've been seeing here. Some differential equations, and that's the within host model that is for flu here. So it's acute virus. So this is probably the virus equation. I is probably in the infected cells. What the H, J, and phi are, I'm not sure. But here H target cells. So they're using different nomenclature. I don't know why. But that's, I guess, an important thing to point out anyway, that if you look at the published literature, you might see a model and you're like, I don't know this model. And then you realize you actually do. They just renamed everything. Unfortunately, there's no real standard. So you just have to carefully read how they define it. And it might be you're actually familiar with the model. It's just you call things differently. Similarly, don't assume just because you've seen P to be the production of virus that they're using that way. Maybe they use P to indicate the rate of death of immune response or infected cells. So always be careful and just don't assume you know what they're doing, because unfortunately there's no standard nomenclature. So they have a nice overview figure here that kind of shows what they're doing. So they have an individual-based model at the population. So individual, in this case, people running around giving each other the flu. And then they can have different characteristics, these individuals. And then on top of that, they're having this model that basically starts running inside an infected person. So once a person got infected, that model here that has virus and infected cells and healthy cells basically in some sense starts running. And then what that does is, depending on how much virus there is predicted by the model that runs inside a person, that's how here infectious and how symptomatic they are on the population level. So what's nice here, let's see, where did I find this? They have a description somewhere. So it starts here where they're basically saying, if you wanted to run this kind of ODE model inside with differential equations inside an agent-based model, it would just take way too long, computationally prohibitive for realistically large simulations. So basically, you still can't really do that thing, especially if you want to simulate more than, let's say, 100 people or maybe 1,000. So it just takes too long. And so then what they're actually doing in their model, even though conceptually they're using that, in practice, they're just then saying, okay, these models produce that kind of curve. And then they're just sort of giving each individual that kind of curve. And then they're tracking the day when they got infected. And then they say, okay, this person is 2 days into their infection, so the virus load is here. And this person is 4 days post-infection, so the virus load is here. By doing that, you don't have to run an explicit model again inside the person, and it runs at a speed that is reasonable. And that's basically how pretty much any of these multi-scale models are doing it these days. You just can't do fully one model inside the other. But so that's an example of a population level model, and inside it a within host model. And then of course they're coupled, and one impacts what's going on at the other level. So that was an individual-based model, and that's basically, all these figures are kind of complex and complicated, so I'm not going to go over it in more detail, but I hope the concept became clear how you can put these models into individuals, but the drawback is still speed, and so for computation, it's often not really done. You make approximations like here. If you have any questions on that, ask or type them in the chat. And in the meantime, I'll pull up the next paper. So this is one of my own papers, and I pulled that up because this is an individual or agent-based model on the within-host level. So what we just saw was agents on the population level, but on the within-host scale, it was still an ODE or compartmental model. So this is now, here's no population level. This is just on the within-host level and an agent-based model. And it actually has both models in it, so that might be interesting. I'll talk you through it real quick. So the idea is, there has been that in the literature, the idea that if a cell is infected, as you learned from Paul, they present pieces of virus potentially on the outside, and then things like CD8 T cells sense it and destroys the cell, and that prevents further production of infected cells. So that's how one mechanism by which infections can be limited. So there was the experimental finding that sometimes infected cells might take pieces of virus and not just put on their own outside, but actually transmit it through small channels to neighboring cells, and then the neighboring cells would put them on the outside. And then, of course, if a T cell came along, it saw it and it thought that cell would be infected and could kill it. Now, that could potentially be good because now this neighboring cell might pretty soon become infected and produce virus. If you kill it before then it can never produce any virus. Of course, the disadvantage is that now you're killing a whole bunch more cells, not just infected cells, but cells that are not infected and might or might not have ever become infected. So the idea is, this, transporting antigen here from one cell, from an infected to an uninfected, is that a good idea or not? And so this was just a paper. There's no data in this paper. It's basically just exploring this idea under what situations might or might not be a good strategy for the immune response. And so we actually started out with the kind of model you've seen before, an ODE model, which is, uninfected cells, infected cells, virus, T cells, and then it also has these what we call bystander cells. These are the cells that get the antigen, put it on the outside, look like they're infected, but they're not actually infected, but they can get killed by T cells. And so here you see a set of equations. It's bigger than we've seen, but in principle, it looks like what you've seen before. And here's also on, this is something very standard in these kind of modeling papers, sort of a table. Let me actually zoom in a bit. A table that lists all the parameters in the model and the starting conditions, hopefully defines them, shows what values they are, either a fixed one. In this case, we kept them fixed, so we didn't do an uncertainty analysis, which I think is okay here because we were mostly conceptually exploring what would happen. But we actually did vary them a bit, as we played with it and saw the overall effect. And then hopefully you'll get a reference that tells you where that number came from. So, every decent modeling paper should have at least something of that form where they tell you where they got the numbers from and the justification for that. And so you need all those to be able to run your model. This wasn't meant to be a very specific, but we chose it sort of in line with flu. We thought of an acute viral infection. And so what you find is when you then run this model is that we just track total virus load and the number of dead cells. And basically the idea is that if you have this transport from one cell to the other, it's just a bad idea. The model would say, no, it's just not a good idea. You produce more virus and you produce more dead cells. So it's not a generally good idea. But of course, this one doesn't have an important feature, and that is spatial structure. And if you have a problem where you think spatial structure is important, then these ODE models are often not so good. And the important part here is that this infected cells doesn't just give that antigen piece to any random cell, but to neighboring cells. And so what that means is the virus is likely coming out of that infected cell and it's diffusing to the neighboring cells. And if there's sort of a gap in between, which we call your fire break, in analogy for how you can fight fires, right? You have the fire burning and then you go next to it and basically try to destroy anything that the fire could catch catch fire on and spread, and then you make a fire break this way. But that is a spatial effect. So for that, this kind of well-mixed model that doesn't have space and it doesn't actually work very well. So then we re-implemented that as an agent-based model. And the main thing we were interested in was that spatial effect. So you've seen that figure before. I showed it as an example. Sort of a 2D grid of uninfected cells. And then you had infected cells, the red ones, virus, the blue, the T cells, and the yellow ones are these bystander cells that you see are basically around the infected cells that have not yet been infected, but that can be recognized by the T cells and killed. And then what we found with the spatial model is that now these gap junctions are actually a good idea, that, that transport of antigen from one cell to the other can actually reduce both virus load and the total number of dead cells you get. So that, I pulled that up because it's an example of a individual-based model. Now, we did not, you could potentially have given every individual cell their slightly own characteristics, like every cell produces virus at a different rate, or the T cells killed at different rates. We did not do that. We basically kept them all the same. So in that sense, we didn't fully exploit what you could do with an agent-based model. The feature we really wanted was that spatial structure, but you could have given every cell here their own characteristics, and every virus as well. Same on the population level model. You could give every individual host characteristics that affect how infectious they are and how long, how much virus they shed and how sick they get based on assumed characteristics. Often that's not done. Often they're still treated all the same. You would basically put the details in there where you care about, and the others you keep simple. So that's an example of a within-host model. And also, I think, maybe nice illustration of sometimes you need those kind of models and the answers are different than what you get from the ODE models. Now, at other times, if that question here wasn't specifically about some spatial feature, it would have probably been okay with the other model. Okay, if you have a question on that, just ask or type them in this. Okay, there's a question. Do the within-host parameters vary a lot between individual hosts? That's a good question. I don't think that is too well known, but they must vary some, right? If you look at, some people get infected and show almost no symptoms, and they have very low viral load, and other people have a very high viral load and get really sick. So that means some underlying things change. Sometimes that could just be random chance, right? As we talked about earlier, if you had a stochastic model and you ran it twice, you could get different outcomes, even though the underlying system is exactly the same. So it's just chance, but it's also quite likely that there's certain features that are different. Some people just have a more robust immune system, whatever that means. So I think these parameters can vary between, individuals if you were to try to simulate different within host individuals. And are there any that tend to be more similar between hosts and any that tend to vary more? I don't think that has really been probed in much detail, which ones might or might not change more. And also you have this, really complex interplay. Sometimes, a 10% change in a parameter might actually have a much bigger impact on the total outcome than a 50% change in another parameter, because the impact of these parameters on the whole model dynamics are not straightforward. They're not linear, right? So some parameters might be much more influential than others. So that also makes it really hard to answer that. Oh, by the way, I might not see any raised hands, so I have said it right. So if you have a question, just shout it out or type it in the chat. Okay, I think we've reached the time, but let me just spend a few more minutes to get through it, and then we'll still take our break. So I want to shift from this one now. So here, basically, we have individual cells and they're infected, but there's no program, so to speak, running inside every cell. So there's just assumed these infected cells, they just pump out virus. And they do that sort of in a black box fashion. So we don't have an internal program running inside those cells. So that would be possible. It's the equivalent to what you just saw. We have a population level model with individual people, and they run a program of infection inside of them. This is one level lower. We have a within host model, and the cells run their own program of virus going in, uncoding, producing proteins, budding from the surface, all that stuff in there. And so there was a question, do these models exist? And they do. So people have modeled sort of within cell dynamics in very much the same way. And so the paper I'm showing you here is a multi-scale model again, but now the two scales are the one we just saw, the individual host, and then inside the cell. And so they have a nice graphic here. So you can see here they have the uninfected cells, infected cells, virus. So this is sort of the one you just saw, that equivalent that I showed you with the green and red and yellow cells. But then instead of just saying, okay, there is an infected cell and this infected cell produces virus, now they're saying, no, let's run an actual detailed program of what goes on inside the cell in the intracellular level. And so that's kind of illustrated here. So you see they have the virus go in, then, producing all kinds of proteins, assembling and then budding. And so the way they incorporate that then on the scale one higher is that they now track the age at which a cell got infected. And then they say, well, it takes some time until a cell gets infected, and then it produces virus, and then at some point it stops and dies. And that is dictated, basically this red curve here, which is the program that happens inside the cell, is now dictated by this process. So exact analogy to what we saw, where we're saying the virus load inside a person and their shedding is dictated by the program that runs inside the person. Here it's now one level down. The program inside a cell, the model inside a cell dictates what's going on in the population level. And so here you see this is the population level. It's a little bit more complicated because they use a partial differential equation, which they do so they can keep track not just of time, but also of age since infection of those infected cells. But you have here the uninfected cells, the infected cells. Here's the virus. So in some sense, it looks more complicated. It is a bit more complicated, but it sort of tracks, broadly speaking, the same quantities that we have already seen. Now, the important thing is that some of these parameters here are determined by what goes on inside the cell. And while they're not showing the model in this paper, here's the model, the intracellular one. You see here sort of this R, the production of virus is determined by what goes inside. And they point to a previous paper they published, so I'm just going to pull it up real quick. So here you see this is now the model here that they did for intracellular, basically where they just did the within cell model, and in that other paper then they connected it. But so here you see again, looks similar, even though it looks messy. A lot of variables that are now tracking certain parts of the within cell production cycle. So these are equations for specific parts of the genes and proteins and certain intermediate products for assembly. And I thought they had it somewhere. Again, here you see it's a big model, so you have a really big table here with all kinds of parameters, their description, their value, and where they got it from. And so you need to do that if you want to model a certain pathogen. In this case, I want to model flu. But so then, this is a within host model. And then this within host model, once you run it, it might then, I'm not sure which one is, but you can run it and then you can look at outcomes that, it's basically total virus produced. I don't know which figures. I haven't read it in a long time, so I'm not sure. But, you get a figure like that then produces, okay, this is total production of virus as a function of time. And that is then what feeds into your model at the next level. Okay, I think that's all. There was one more question about, if you have any questions, type them in the chat or unmute. I'll pull up one real quick. there was about reassortant. I'm not too familiar with these kind of dynamic models about reassortant, but Katja Kölle, who came up earlier for the dengue model, she's also been working some on that kind of question, broadly speaking. So here's a paper that she and others did. It uses modeling, but it doesn't use the kind of modeling we've been looking at. So if you look at the models are all here in the back. So you see single locus model. So they call it model, and it is a model, but it's not these kind of dynamic mechanistic models that we've looked at. These are more the kind of models, you know, from population genetics that are trying to capture processes. So, you know, they're, in some sense, they're one level more detailed than just a classical regression model that just says, let's see if there's a pattern in the data. Let's see if a linear model can explain it. So there's some kind of notion of underlying, you know, what might be going on, but very abstract. For instance, you know, assuming there's exponential growth in the population or there's constant population size. So those are these basic population genetic models. And so they're fitting that kind of model to it. She has a few other papers where she looks at that question a little bit, but, you know, basically multi-infection of cells instead of a cell being just singly infected multiple times, which of course you need to get reassortant going, but not there directly. But, you know, that, you know, looking up her work is a good starting point for that. And I think these were all the papers I was going to pull up. I hope I covered most of the questions that people had. If not, let me know. Post it in the chat or unmute or Slack. Yeah.
I have a quick question about the multi-scale models. So I'm curious how like time would work in those. I'm thinking especially like back towards that first paper you shared where they're modeling like within a person and then between people. I can imagine that like within a person, you might want to model on like at a scale of time of either like, I don't know, minutes or hours, because like things can happen much more quickly within a host, but like between people, like hours is like not worth it. You might be like more on a scale of days or weeks, or how does that work in those models typically?
Yeah, so that is really what you touch on there. That is the problem why they're computationally so expensive. So if you, even though you might be able to get away of taking time steps of days in your population level model, that's not detailed enough for you within host model. So basically what you have to do is you have to sort of pick the unit of the process that's the fastest. So in this case, you know, you might have to run it in time steps of hours if you wanted to have a fairly detailed within host model run there. And that is just not feasible. And that's why you cannot do that. And that's why they're doing this approach here where they're saying, okay, we're running this kind of model, but then we're just using the model to extract these curves. And then we're basically hard coding these curves into individuals. And then we jump sort of in days. And then we can say, okay, if you've been infected for 2 days, then you have this much virus. If you're infected for 5 days, you have this much virus. So then you can go back to the bigger scale and run it in reasonable units. But yeah, this is a general problem. And anyone who's done these kind of models in chemistry, it's a big problem there too, because if you have, and so there's lots of work there. If you have, you know, a lot of chemicals that are reacting, you know, some of them might just do 1 reaction per minute, while others do like 100,000 reactions per minute because there are many more of them. And then you would actually have to adjust to the fastest one, and that often makes things slow too. So there are computational tricks you can do, but in the end, at some point, you just have to make certain approximations to make things run in a reasonable amount of time.
Got it. Thank you.